Okay, we are good to go with our YouTube live stream, and I want to welcome back Adam Shillard, the original instigator of cell migration seminars. He is guest hosting today. Ankita is in India getting her visa situation worked out. So we're very happy to have him back, and he'll be helping out. So Adam, I'll let you run things in the beginning here.
Thanks, Jen. So let's get cracking. To start with, we have a talk from two graduate students, José Almeida and Jairaj Mathur, sorry if I'm mispronouncing that, who are graduate students in Amit Pathak's lab at Washington University. And they are going to tell us about mechanically primed cells encode matrix memory in fibrous environments for persistent invasion. Over to you guys.
Thank you. Here, let me share my screen. Here, everything good? Yeah, cool. Well, yeah, thank you for the introduction. I'm Jose, this is Jay. And yeah, we work in the Pathak lab here at Washington University. And today we'll talk to you about, the title's a little different, you know, it changes, fluctuates it over time while I submitted the abstract. But the title now is Cellular Mechanical Memory Remodels Collagen Matrices for Cross-Environment Collective Cell Invasion. It is the same topic. So let's talk a little bit about the background. So cells obviously interact with a lot of mechanical gradients as they do different processes. For example, in cancer, you have invasive fronts where you see this stiffness map on the left, where the redder it is, the stiffer it is. And then the surrounding environment is actually softer than what it is in the tumor environment. And then during breast tissue cancer development, you have the tissue becomes denser and more fibrotic. So cells have to overcome these different environments and these mechanical gradients that are present. In the similar case, when it comes to wound healing and repair, epithelial cells have to go from a healthy outside tissue, which is usually softer, to a fibrotic site, which is usually stiffer. And in neurocrest development for these complex organs, organizations, you have to also transverse to different environments. So these cells go and move through these heterogeneous environments around their ECM, and they do this through actin and focal adhesion signaling. But we're mostly curious about how the idea of memory and plasticity is impacting cell migration. So like as a lot of us know here, obviously, but cells stiff have a very specific phenotypical aspect and soft as well. And when it comes to plasticity, where the cells are able to adapt, they have a fast adaptation. If they were transversing from a soft environment from a stiff environment to a soft, you actually have an instant adaptation. But when that doesn't happen, this is what we kind of term as mechanical memory, where there's information retained from a previous environment as they go to a new environment. So from our lab, it's one of many works, but from our lab, in 2017, we published this work where we found that there's a stiffness, a memory dependent, some migration through YAP enforces. So YAP is part of this player that allows for this retention, this delay in plasticity. And when you deplete it, essentially that plasticity overcomes and it becomes fast again. And for us, what inspired me from this work, and also Jay, was that we wanted to look at the three-dimensional aspect of this, specifically in a fibrotic environment where, as from all the examples that I mentioned above, all of this is like in a three-dimensional environment, fibrotic like collagen. And also if a previous environment, and information of a previous environment can actually affect a new, different environment, right? So in order to answer this question, we developed an in vitro model that we use polyacrylamide gels, where we prime.
Can I just ask you to, I really don't understand what you mean by memory. And if your whole talk is about this, maybe it would be good to just clear that up a little bit. I don't understand what you mean.
Yeah, no, so.
Sorry to interrupt, but I figure probably other people don't understand what you mean either.
No worries, it's perfectly fine. So
thank you.
Essentially, I think I'm gonna maybe switch the term from memory for just information, right? retention of information that they had from their previous environment, right? So if they're on a stiff and they're invading very fast, then that information still retains as they go to a soft environment.
What do you mean by information? I mean, I understand what memory
So,
means, but I don't understand what it means in a cellular, in a, you know, molecular context.
you know, it could be like different degrees. So what would you kind of describe it, Jay?
It could be like I think right now what we kind of mean is like the behavior on stiff and behavior on soft is like influenced by their past environment. So as they go from the stiff environment to soft environment, they continue to migrate fast. Like this was a paper.
Okay. Okay. Thank you. I get it. Thank you.
Thank you for that question. I think it clears up a lot of stuff. So, so going back to this, we use a polyacrylamide gel. We prime them on a soft and stiff environment. Our soft is 0.8 kPa and our stiff is 16 kPa. We use MCF10As, which are normal breast epithelial cells. We then, after those 5 days, we encapsulated the cells in collagen, 2.3 milligram per milliliter concentration, and we let them invade for 3 days. And what we found was that, excuse me, we found that the collagen, there was higher collagen alignment for the stiff prime cells compared to the soft prime cells, and due to potentially a higher actin bundling, where the cells are engaged in the collagen more compared to the soft prime cells. So there was a tension being developed after those 3 days of fixing. And when we did a trypsinization experiment, we found that there's a high degree of tension in the collagen after the cells release. They release it after 3 days. So we wanted to look at the overall invasion. When we found the overall invasion of this after those 3 days, we saw that there was a higher stiffness higher invasion on the stiff prime cells, and there's a lower invasion for soft prime cells. There's higher amount of cells present, and there's a lower amount of cells present in the soft prime cells. So this is over the track of those 3 days, and he made us curious to see, want to do a temporal analysis of what was happening. So next, we'll show you a video about essentially what we saw. On the red rectangle is just the stiff prime cells. On the blue is the soft. So here is stiff prime cells that are GFP labeled. As you can see, the collagen is mixed with beads, and there's a large pulling and deformation happening. And once a band is formed, there's invasion occurring for the stiff prime. So then when we look at the soft, this deformation doesn't happen. The deformation is a lot less, and overall, the invasion of the cells actually decreases. When we quantify this, and I want to point out at the 26-hour time point, we see that we use PIB to track the bead deformation over time. So that how much they're deforming, so the deformation rate of the collagen. And we found that there's actually high deformation where the collagen becomes under tension. There's a formation of a band of high collagen intensity, and then the cells migrate afterwards. So when we look deeper into this, we find a couple of things. So stiff prime cells invade faster than soft prime cells. And then there's an accumulation of collagen fiber and densification of the ACM prior to their invasion. And this accumulation possibly acts as an anchor that develops the tension that is utilized for their future invasion. So here we have cells that come from one environment that have a information or some phenotypical aspect of some degree of this information of the previous matrix as they go into a new one. And this leads to remodeling of the collagen matrix. And then this remodeling is allowing the cells to aid their future invasion. And in order to look at this in a more mechanistical way, we wanted to develop a computational model that Jay was in charge of, and he'll talk to you a little bit about it.
So to understand this priming dependent cell migration, we start with the basic model of how cell migrates. So there are 3 key steps involved. First, cell extends an outward protrusion. There are adhesions formed with the ECM, and there's a retraction of the rear adhesions. And each of these steps have an energetic cost associated with them. And we can look at this migration as the protrusions that the cell is trying to make minus the resistance offered by the adhesions. And we know that protrusions, without any protrusions or without any turnover of these adhesions, there is no cell motility. Looking at 3D environments, this migration becomes protrusions minus resistance plus remodeling. And where this resistance is not due to only adhesions, but it's also due to your bulk collagen steric hindrance. And your remodeling corresponds to collagen alignment, proteolysis, collagen deposition, etc. So based on this, we formulate our energy equation for the cell as having 3 components: H bulk, which is the energy required to increase cell volume; H adhesions, which is energy required to make adhesions with the ECM; and H protrusions is the energy required to make stable adhesions, to make stable protrusions. And the cell will only be able to migrate forwards if the net change in energy is less than 0. We propose that cells utilize collagen remodeling as a way to lower the net energy barrier. So we formulate that delta HP, which we call as our mechanoactivated migration potential, mu, has 3 components. That it has the current environment sensing, which we call as mechanoactivation. So that is the signal to the cells from wherever they are right now, minus gamma, which is the resistance they face from the ECM as they enter into it, plus collagen remodeling alpha. And I'll represent the tension in the ECM as alpha bar, which is the net collagen remodeling happening in the field of view. So cells remodel the collagen, and they're able to lower this energy barrier and lower delta H overall, and able to invade further. In the absence of collagen remodeling, the energy barrier remains high, and your overall invasion stays low. We propose that collagen remodeling is a combination of 2 signals. One is the memory signal, and the other comes from direct mechanosensing phi. We propose that both of these, phi and psi, have varying kinetics. So phi, which is your direct mechanosensing, has a faster kinetics because it is rooted in adhesion formation. And your cellular memory, which is based on your YAP kinetics, as it has been shown in our previous work, it has like much lower kinetics compared to mechanosensing. So combination of phi and psi inform your collagen remodeling. So with this model, we simulate what happens when stiff prime cells migrate into collagen of density 2.3 mg per ml. And when we track phi and psi, which is our mechanosensing and cellular memory, we see that the mechanosensing signal quickly adapts to this new softer collagen environment. But cellular memory persists for a while before adapting. During this time, we see that the tension in the ECM increases, which helps mu, your mechanoactivated migration potential, to increase. Compared to the soft prime case, both of these signals are low to begin with. So when they come into the new environment, there is no rise in tension as it happened in the stiff prime case. And your mu, which is your net migration potential, it stays low. When you look at the spatial collagen remodeling maps, so these are the simulations and they run over 3 days. You see that in the stiff prime case, cells are able to create this dense environment around the edge of the gel, which we call as the collagen anchoring. And they're able to utilize this collagen anchoring and create tension in the ECM, and they're able to invade further into the ECM. This anchor formation does not happen in soft prime case, and consequently, the net cell invasion rates for the 2 cases are quite different. Next, we wanted to ask, what is the role of cellular memory in this process? Is it just that because stiff prime and soft prime cells are in different environments to begin with, and their initial levels of phi and psi are different, is that's what causing this varying collagen remodeling in these cases? Or does the slow decay of mechanical memory, does it truly have a role to play? So we simulated the case where stiff prime cells, they go into collagen environment, but their YAP signal, the psi, that adapts much quickly. So, and we control that by our parameter zeta. So we see that in the YAP depletion case, there is no collagen anchor formation that happens in the control case. And we also see that the tension in the ECM, which is on the right here, in the right plot, that is also lower compared to the control case. And consequently, your net cell invasion rates for YAP depletion are also lower. Thus, the slow decay of mechanical memory is required to, it is important for cells to be able to persistently invade into the ECM. Next, we also wanted to see what is the role of the propagation of this tension throughout the ECM. And we do this via parameter n, where if you increase n, the distance over which the tension propagates reduces. And we see that there is lower collagen anchor formation, lower tension in the ECM, and your net cell invasion rates are also lower if the tension cannot propagate throughout the ECM. We also wanted to see what is the role of collagen, the rate of collagen remodeling in this process of cell invasion. And we systematically varied our parameter R alpha, which controls how quickly cells are able to remodel the ECM. And as we systematically lower R alpha, we see that the tension in the ECM goes down. And for the lowest value of R alpha, there is no collagen anchor formation at all. And consequently, your net cell invasion rates also decrease. So in conclusion, as cells move from one matrix to the other, there are three key steps involved here. The cellular mechanical memory is encoded into the matrix via forces. The matrix memory is stored in terms of collagen remodeling, which is fiber alignment and tension. And cells are able to extract this matrix memory to persistently invade. The model also suggests these two cellular processes, phi and psi, they occur at different rates. Mechanosensing happens quite fast, which is via adhesions, but the cellular memory psi slowly adapts and which gets stored into the matrix as alpha. Next, we wanted to test out a couple of these predictions by disrupting the cell to matrix memory transfer and seeing the invasion phenotypes.
So in order to do that, we depleted the system with YAP. Lack of YAP essentially showed, as you can see from the graphs above, where the dotted line represents the control. For stiff prime cells and soft prime cells, their deformation became noisy and chaotic. And overall, their invasion, there's no significant difference between their invasion of stiff and soft. So now we're going to take a little step back from this and just talk about the environment overall, right? We mess with the cell's ability for the cell matrix transfer, but the cell aspect. Now we're going to look into the matrix itself. And we wanted to see if priming can actually affect dense and stiff collagen environments. So in order to do that, we used different collagen concentrations. We used 3.1 milligrams per milliliter, and then we used our control concentration, but that is cross-linked. What we found was that we used riboflavin to cross-link this. Our control concentration is the same amount of collagen. It just the fibers are more cross-linked. And as you can see, it's a lot more stiffer. And we found that the fiber alignment is independent for the dense collagen case, but it is priming dependent for the stiff collagen case. In addition, when we look at their ability to deform the matrix, these are chimeographs essentially showing representing the band that you saw before in the previous slide, just a different illustration of them, is that for the control, you have a high band formation. For base, this is the collagen bundling based on the bead intensity. And then for 3.1, with the band is a lot lower, but for the 2.3 crossing concentration is still high, as we've seen from the control. When we look at the priming advantage, which is the difference of invasion rate for stiff and soft, we see that the priming advantage remains, meaning that over time, the cells are invading faster and more consistently than their soft counterparts. Essentially meaning that this priming is persisting in this dense collagen-rich environments and cross-linked environments. And here we have results from the simulation that matches the results that we gathered experimentally. Like Jay mentioned, we wanted to look at the tension propagation, the force propagation that was happening. So in order to do that experimentally, we used laser ablation, where we cut the fibers before the cells invaded, the stiff prime cells were unable to make this broad movement that you saw from the videos from before. And their invasion decreased over time, meaning that the invasion rate declines because the lack of this long distance force propagation from the collagen. To look at also the remodeling, so their cells' ability to actually remodel the matrix, we use LOX inhibitor. And through LOX inhibitor, we found that there was a massive decrease in their ability to deform the gel and also their ability to invade for both cases. And overall, this is the conclusions for the experimental model and for the experimental side work and the experimental model work is that stiff arm cells apply higher forces and align the collagen, but it's in two phases. So the cells reach the collagen slowly. Then once they remodel the collagen, they're able to they essentially transfer the matrix memory. The cells then utilize this memory for their ability to invade further. And it requires two different sides. In the ECM side, it requires a LOX-based cross-linking, and it also requires a long-distance force propagation. And in the cell side, it requires a YAP-based mechanical memory, and also cell forces via cell-to-cell junctions to have a collective force, coordinated force development. And with that, I'd like to thank our lab members, Dr. Pathak, our funding sources, and these are our contact informations. We're looking for postdocs, and we're open for any feedback, and welcome any opportunities you might have. Thank you.
Thanks, guys. We've got some questions already. Can I just kick off with just one of mine, just for clarity? So, for that, I mean, this is kind of related to what Claire was asking earlier about what the memory means. So, is it cells that are on a stiff environment, they have high nuclear YAP, and then the targets of that YAP-dependent transcription are the transcribing things that enable the cell to then generate higher forces? Is that correct?
Yeah, potentially, right? So like when we did the PCR for them, we saw a higher row and rack activity for the stiff prime cells specifically compared to the soft. So yeah, I think the app has that influence over it because they're like, essentially it is transcribing stuff to help them with their ability to generate forces. Anything you want to add to?
Yeah, thank you.
Okay, great. We have a number of questions from Sonali Sengupta. Sonali, if you want to ask them yourself, feel free to unmute. Otherwise, I'll just go for it. So the first one for Jose, does the memory depend on prior environmental interactions? Are these prior interactions encoded in the cell matrix interactions? And is there an underlying code involved in this memory as compared to neural memory of engrams?
Well, okay, that's a really good question. Very deep. So I think the encoding for our side is mainly in the matrix, right? So there's like this ability for the cells to actually remodel the matrix, and that's the encoding that happens in the ECM as they invade outwards. I guess I got to look a little more into it in the question about like how it relates to neurological ways that you mentioned. But yeah, excellent question. I'll have a lot to think about. Thank you for the question.
Great. Yeah, Adam, do you want to take Jayadi's question?
Yeah, it's how were the specific time points decided for the priming of the cells?
We decided 5 days. In the previous paper, the longer you prime them, the more that behavior remained. But we decided that 5 days was like the number we wanted to keep it at, essentially.
Consistent.
Yeah, it's a consistent number that we had from previous work. So we kind of just maintained that. Yeah.
Great. Another question from Sonali asking, what's the correlation between the YAP and collagen?
So for YAP and collagen, so like in what extent? Like I guess I need a little more explanation. like for the results of like their ability to deform or
Sonali, do you
Yeah,
want to unmute?
if you want to clarify, that was
So
during your talk. Go ahead.
Yeah, so thank you, Jennifer. So YAP is a part of a signaling pathway, right? YAP hyposignaling, right? So it's a part of an oncogene tumor suppressor axis. So it's intracellular, while collagen is extracellular. So how does the intracellular signaling translate to cell matrix extracellular environment interactions in the context of memory?
Okay, so from what we have seen in the previous work for some, I guess 2017 and now, YAP does have influences or actin forces, higher development. So that's how like you go from internal to like external, essentially. How it might affect cross-linking and anything else, that I think we need to look more into it. And I think it's an open question for everybody.
Sure, thank you.
I mean, related on the cross-linking from Maxime, can you elaborate on how the cells are able to modify the alignment of the collagen fibers? Is it relying on the cross-linking?
I think part of it is relying on the cross-linking, yeah, because of the dense collagen bundling that we see at the beginning from like that dense band. So that they have to be able to remodel it, cross-link it, and then be able to have the tension is essentially from them pulling and actually making it like an anchor. That's what we're speculating. And also with the Lux results and even through the experimental model, we saw that it is important to have the cross-linking there for them to be able to have that. But it's a mixture of forces and cross-linking, I would say.
All right, great, thank you. We have a question from Farrell asking if you can just repeat or sort of clarify about how you primed your cells.
Yeah, so like we essentially seeded the cells and left them there for 5 days. Essentially, they grew, they expanded in the PHL, and then they were encapsulated in collagen. So the priming is essentially letting them be there for a long amount of time.
So
yeah, I have a
oh, sorry, go for it.
Oh, go ahead. No, you can go, Adam.
I was going to ask about MMPs. Do you know if there's the differential degradation of the matrix when you in the differently primed cells?
So we looked a little bit into that. I couldn't confirm, honestly. I think it's something we potentially have to look into it, but that's a really good idea. Yeah.
All right, thank you. From Anna Pasapera, if you want to go ahead and ask.
Yeah, hi. It's an interesting work. So I was wondering the correlation between migration and YAP. Have you ever think about using mutants of YAP that cannot go to the nucleus so you can separate the transcriptional regulation versus YAP interacting in signaling pathways with raw GTPases and things like that to see if you require transcriptional activation of YAP when it's translocated to the nucleus?
That actually, no, I think that's a really good thing, and I'm gonna look into that because I had no idea you can actually do that. That's actually really cool. Yeah, thank you. And I wish I could answer the question, but yeah, we look into that.
All right, great. We'll just take, I guess, this last question since we'll want to move on. But Farrell is asking, as a follow-up, did you have them cultured on the gels that had different stiffnesses before moving them to collagen, or is it just the length of the culture that varied?
So we seeded the cells on the stiff and soft, and then we cultured them for 5 days straight. And then we just moved the gel into the collagen. The entire gel is encapsulated in the collagen. Yeah.
I have a follow-up to that myself. So do you think that this memory that you see of what the cells were initially cultured on, this difference between soft and stiff, how much of that do you think is really required to be specifically signaling from collagen versus just the fact that just the stiffness? Because I'm thinking that all of the cells that we culture are essentially stiff primed, right? Because we're all culturing cells on tissue culture plastic, which is incredibly stiff. So are we sort of just default working with stiff primed cells in the lab without really thinking about that?
I would say, yeah, I think it has an influence depending on what you want to do. Yeah, and that's part of another reason why we prime them for so long, is the idea that like they were on a flash for a decent amount of time, and when we take them out, they should be there for also a decent amount of time to get rid of any of that potential like TC plastic feedback or something that could potentially happen. Yeah.
Yeah, and I think like as per your question is like, how important is the memory from the cells? I think that's what the model shows, that like even if the cells are stiff primed, but they do not have the cellular memory, they are not able to persistently invade into the collagen, because they cannot like pull on the collagen, they cannot like form the collagen anchors and increase tension in the ECM, which eventually allows them to invade further.
Yeah, so it would be interesting to compare stiff versus soft substrates that with ones that were made from collagen and the ones that weren't made from collagen to expand and tease out a little bit more of where the actual matrix alignment is required versus just stiffness versus softness. Okay, I'm gonna, if there's any more questions, people feel free to ask in the chat, and Jose and Jay will can answer there. With that, I'll thank both of you for a really fantastic talk, and I'm glad that you were able to do this joint format. I think that's really nice and worked really well. So thank you so much for your presentation and for the initial abstract that you submitted, which was fantastic. With that, I'll ask you to stop sharing your screen, and our second speaker can putting hers up. So we are delighted to welcome Dr. Marion Baraban. Marion's a postdoctoral researcher at the Institut de Biologie in Paris, and she's working on formation of the olfactory orifice in zebrafish in the group of Marie Bro. Marion was also previously a postdoctoral fellow in the group of David Lyons, where she worked on the interaction between neurons and glial cells in zebrafish. Again, she submitted an abstract that was really fantastic, and we're so excited to hear about her work today. With that, Marion, the floor is yours. Thank you.
Okay, thank you very much. So first, I would like to thank the organizer to give me the opportunity to talk today. And I'm working, as you say, in Paris in the developmental biology laboratory. And my work is focused on the olfactory orifice, which are formed by the interaction between two cell types, the sensory neurons and the skin. So first, as a general introduction, normally epithelium layer has a barrier function, but recently some work have shown that disruption of epithelium layer can occur during physiological development. And here I will present two examples. The first one is concerning the Drosophila leg imaginal disc, where the peripodial layer which envelop the leg will be removed and facilitate the growth of the leg. The second example is the maturation of the Drosophila oocyte, where a transient and local opening will occur at the tricellular junction of the follicle epithelium, and that would be important for the import of your protein in the egg. So I have shown you two striking examples where the loss of the integrity between skin cells is essential for the development in those two systems. And it's also important for other organs, such as the sensory organ, because it makes sense for the sensory function to have a gap within the skin in order to access to the sensory stimuli. So then the sensory neuron would be in contact with the sensory stimuli. And it's really important to have this gap within the skin. So actually, the system we are using in the lab, which is the olfactory system of the zebrafish embryo, is well suited to study such questions because at early developmental stages, the sensory neurons are completely covered by an intact layer of skin. And then the formation of an orifice within the skin will reveal those neurons, and then they would be in contact with the outside and able to detect the odors. So really, in this model, the formation of a gap is essential in order to allow the function of the sensory organ. So my project is to look at the cellular and molecular events that occur for the formation of this orifice. So first, I would like to describe the system because it was not well described previously by beginning of looking at the before the orifice opening. So how this system is organized. So we look at the first component, which is the skin. And normally the skin in zebrafish is composed in three different layers. So the peridermal cells are the more superficial layer. They cover the basal cells, which are laid on the basal membrane. And actually, we look at the peridermal cells. So above the olfactory placode. So the olfactory placode is this organ that will contain the sensory neuron. So above the olfactory placode, the peridermal cells, before the formation of the orifice, form an intact monolayer that you see there. So what about the two other components, the basal cells and the basal membrane? We were surprised to see that both basal cells and basal membrane are completely absent above the olfactory placode. So the only remaining cell type of the skin, which is present above the olfactory placode, is the peridermal cells. So for now on, I will talk about them as skin cells because they are the only component of the skin. So concerning the other component of the system, the neurons, how are they organized? So they are organized in a rosette of neurons that you can see here. And actually the cell body is extending a dendrite, and all those dendrites are forming a little cavity, the rosette cavity. And very strikingly, just before the opening of the orifice, we found out that there are huge deformation of the skin, as you can appreciate here, and I will comment later on about those deformations. And those deformations are specifically happening above the cavity. So that was a striking result, showing a huge interaction between those two cell types, the skin and the sensory neurons. And actually about the cavity, there are enrichment of several components, including the ZO1 marker, which is also called the tight junction protein 1, which accumulate at the tip of the sensory neurons and allow to see the shape of the cavity. So altogether, we have a system where a monolayer of skin cells is covering the olfactory placode, and within the olfactory placode, a rosette of neurons where the center is forming a cavity is present. So that was the system before the opening of the orifice. So how does the olfactory orifice open? So I performed some live imaging using some transgenic line where the skin membrane was labeled just above the olfactory placode. And I guess you can appreciate that the olfactory placode formed by a local loss of contact between skin cells and the cell division and the cell death were not required for the formation of this orifice. So what about the deformation of those skin cells that are at the edge of the forming orifice? So I analyzed the shape and the thickness of those cells and found that they are modified in comparison to the other cells that are far away from the orifice. So they are modified because they look more round and they have lost their stereotypical thin appearance as an epithelium. They also have lost their honeycomb-like shape, meaning that they are specifically deformed. So those cells are deformed. So what about the localization of this orifice? Where does the orifice form? So we look by co-labeling the two cell population of interest and found out that olfactory orifices always open above the center of the rosette of neurons. So that was another evidence that the rosette of neurons seems determinant, and specifically the center of this rosette, the cavity, will trigger the opening of the orifice. So in addition, we get further information about zooming at the different elements of the cavity, which is the component of the neuronal rosette, versus the skin component with the orifices, and found out that when a cavity is in contact with an open orifice, this cavity is bigger than the one in embryo where the cavity was associated with no orifices. In addition, we found a correlation between those two structures, the orifice and the cavity, because as you can see, we observe a correlation of the area of the cavity and the orifices, and also about the shape of those two structures. So that was more evidence suggesting a really important interaction between those two cell types, and the fact that the cavity might be essential for the opening of the orifice. So in order to test this hypothesis, we want to disorganize the rosette and see the consequences on the forming orifice. So we would like to take advantage of a perturbation of a signaling pathway, which is the CXCL12A chemotactic cue. So just to mention first that this signaling pathway is only active in neurons because the receptor of this cue is absent in the skin and specifically expressed in neurons. And very interestingly, previously, some studies have shown that this cue is essential for the early step of morphogenesis of the placode. So we wonder if this change of the organization of the placode could lead to some disorganization of the rosette. So to test that, we look by downregulation of the cue, what was the effect on the formation of the rosette. So here you see in control situation, you have a round rosette of neurons Whereas when we injected the morphine, we observe a variety of phenotype with a smaller rosette, elongated rosette. That suggested that there were disorganized rosette of neurons in such condition. So we now want to address what was the consequences on the cavity, so the center of the rosette, in such disturbed, disorganized rosette. And for that, we used the ZO1 marker that I presented previously. So normally in the control situation, you have a round shape of the cavity, whereas in the morphine situation, we observe a variety of shape of the cavity with a smaller cavity, deformed cavity, and even sometimes we end up with multiple cavity, meaning that we have several rosettes within an olfactory placode. So we now want that to address the consequences of this disorganized cavity on the orifice. So for that, we look at the cell shape, the shape of the skin cells using the ZO1 staining, and that will allow to see the orifices. And basically, we were amazed to see that both the cavity and the orifices always match in area, localization, and shape. So that was really for us confirming this hypothesis that the neurons are instructing the skin to open a specific location. And furthermore, this example of multiple cavity related with multiple orifices was an evidence of the necessity of the presence of neurons to open the orifice. So altogether, we think that the center of the rosette of neurons is opening the olfactory orifice within the skin. So in order to go further, we want to decipher the role of mechanical tension in our system. So for that, we look at the molecules that are involved in the generation of forces in our system. And especially, we saw a huge enrichment of myosin at the tip of the dendrite of the sensory neurons. So we then look at the dynamic of this myosin in live. So here it's a movie where you see the skin membrane labeled just above the olfactory placode, and the specific expression of myosin reporter, where basically you see a huge accumulation of the myosin at the tip of the dendrite where the cavity is located. And I guess I will play the movie. You would appreciate that this ring of myosin is increasing in size during the opening of the orifice. So there is a huge increasement of the myosin ring during the opening of the orifice. So that was suggests that the neurons might be important in order to trigger the opening of the orifice through mechanical forces. So we then want to address the importance of myosin activity in this context. So we want to perturb, I forget so basically we just, about those movie, we quantified the increase in the size of the myosin ring during the opening of the orifice. And we also found that there is a correlation between those two structures of the cavity and the orifice over time. So all those data about myosin activity lead us to suggest that myosin is really important for the formation of the orifices. So we now want to inhibit the myosin activity and see the consequences on the orifice formation. So we apply blebistatin during the process of orifice formation, and strikingly, we found that in all the treated embryos, there were no orifice formed. So leading to this idea that myosin activity inhibition will completely prevent the formation of the orifice. And then myosin is really important for the formation of this orifice. So we then wonder if the neurons might be under mechanical tension. So using a laser ablation technique in collaboration with Isabelle Bonnet at Curie, we severed the dendrite and observed the recoil effect. And we found that in most of the cases, there is a recoil effect, meaning that the dendrite of the sensory neurons are under mechanical tension. So altogether, that leads us to propose this new model where the sensory neurons exert pulling forces on the skin that will allow the opening of the olfactory orifice, which expose then the neurons to the outside, and those neurons would be able to detect the odor cues. So for the moment, we are investigating the role of adhesion molecule in the formation of the orifice. So I would like to thank all the people involved in that work and everybody for your attention. And I will be happy to answer your question.
Fantastic. That is really, really cool. I just want to start out with one question that I don't know, this might be a silly question, but when I see a ring of myosin, my assumption is that that is going to contract and get smaller. And you showed us a ring of myosin that's getting bigger. And so I'm just wondering how you sort of square that in your head. And is it just because the neurons are pulling and the myosin is resisting that deformation? Or physically, what are you thinking is happening there?
So it's true that it's not a conventional ring of myosin. What we think from like a molecular point of view is more like a radial oriented part of myosin. Instead of being like a circle, it might be more an apposition of myosin structure that are going in the opposite way, but behave together by cohesive molecules. For example, some membrane-targeted protein structure, things like that. So in the way, I think it's the key point to address such to have an overview of this process in 3D to really understand how this structure evolves.
Great, that's really beautiful. Thank you. Adam had to leave. There's a fire alarm in his building, so it's just going to be me going through the questions. So from Natalie Hussein, saying impressive movies, agreed. Very impressive, very insightful presentation, Marion. Thank you. And she asks if anything is known about the formation of these underlying cavities prior to the opening of the olfactory orifices. What are the signaling pathways involved? More specifically, in the formation of the olfactory placodes, when the neuronal rosettes are formed, is the embryonic epidermis already missing the basal cells and the basement membrane?
So there are like multiple things to answer to those questions. So the first one about the formation of the cavity itself, it has not been yet looked at. We have like some evidence of the biology of how it works. We still don't completely understand its it would be another part of the story, I guess, because it's a process that might be due to chemical cues that will need to be investigated. About this second question and the absence of basal cells, I look at very early stages during the formation of the olfactory placode, and actually I found that they are already missing, or they are excluded from this particular region at very early stages. So it would be also something to look at. What are the signals required for this exclusion? Is it an exclusion? Is it an absence of migration of those cells? Because we know that those cells at early stages, they are having a migration behavior. So yeah, it's also another things to investigate.
Right, fantastic. Thank you for that answer. A question from Hani Suleiman saying, great talk. Are the neurons underneath anchored to a basement membrane to be able to pull on the ring?
So that's really, we think that, so we check for basement membrane, and they seems all excluded from this region of the cavity. So that was really striking to us to see this absence of basement membrane. But so we think that really those molecules are completely absent. Collagen, laminin. So yeah, it's independent. It's ECM free.
Okay, great. We just have some comments. Beautiful talk from Carrie and Neeson, and Nathalie says, thank you, and looking forward to the next chapter. From Lara C, saying, amazing talk. Do you know if there are any other cell types involved in the process? For example, glial cells surrounding the neurons. And then also what happens to the epidermal basal cells? Are they dying? Which I think you were getting at before.
Yeah. So about the other cells involved, we don't think they are like other cells. There might be like different population within the sensory neurons, but really like when we use co-labeling transgenic line, we manage to reach a full we managed to see all the cells labeled with that. At this stage, the glial cells are not yet differentiated, so they are not present at all. And about the apoptotic cells, in which structure you said?
And the basal epidermis?
Ah, oui. Yeah. So about this early process of exclusion, absence of migration, we didn't see any like apoptotic things at the stage I'm looking at, but it would need to be looked at for the early developmental stages. Yeah, it's something to look at.
Fantastic. I think that's all the questions from the audience, unless anyone wants to pop in while we're closing up in the next minute. I was just wondering, is this a mechanism, anything like this has been observed in other sort of placodal populations, or is this the first time that we're really seeing this kind of, neuronal pulling and signaling from the neurons to say, open up here? And yeah.
So there is one system that shares some similitude with this organ, and it's also the Neuromast System. So the Neuromast System are the organ that sense the fluid in the fish, and they are like really striking analogy between the exciting cells that look like our neurons and the skin interaction. But it's much more local and there are less cells involved. And when you look, for example, some EM overview of this system, you can see some deformation of the skin, which might suggest some analogy with the things we are looking at. But again, it has not been investigated yet.
That's really wild. It's really, really beautiful work. All right. And again, thank you so much for the abstract you submitted. It was fantastic. I'm glad we got to have you today. With that, I'm going to say bye to everyone on YouTube. I'm going to end